Hello, everyone. My name is Eric Jamois, Director of Business Development at Simulations Plus. On behalf of Simulations Plus, I welcome you to today's webinar, which will discuss and demonstrate the new features in ADMET Predictor 9.5. This webinar will be presented by David Miller, Director of ADMET Chem Informatics. Joining David as panelists are Michael Lawless, Senior Principal Scientist, and Bob Clark, Senior Research Fellow. An opportunity to ask questions will follow the presentation. You may either send your questions using the questions pane on your control panel, or you may ask your questions directly using the hand raising icon. This webinar is being recorded for playback at our website, www.simulations-plus.com. Also, upon request, the presentation slides will be made available and shared with colleagues. I'm now going to hand over the controls to David Miller, and thank you for joining us this morning.
Okay, thank you very much, Eric, and thank you everyone for attending today. The seminar, as Eric said, will be about the new features in ADMET Predictor version 9.5. I will first give a brief introduction to ADMET Predictor. I'll describe most of the new features that we've added to the version, and then we'll give a software demonstration. I would like to spend most of the time on the demonstration portion, so I will be brief with the first two bullets. But there are some additional slides that will be available in the downloadable version of the presentation. Okay, so what is ADMET Predictor? The primary functionality is property prediction. Currently, the software predicts more than 140 ADMET properties from chemical structure. It also identifies potential ADMET liabilities in the form of numeric scores that we call risks. HTPK stands for high throughput pharmacokinetics. That's a relatively new module. It lets you predict fraction absorbed and bioavailable, as well as other PK parameters using a virtual human or rat simulation. The ADMET Modeler module lets you build your own models using our advanced molecular and atomic descriptors. This is also the modeling engine used internally by Simulations Plus for our commercial models. The MedChem Studio module lets you prioritize lead series, discover SAR trends, and design novel compounds. The software is divided into modules, and the four specifically devoted to prediction include the PCB, metabolism, toxicity, and HTPK modules. In the additional slide section, we have more comprehensive information about these modules, so I'll just briefly outline each of them here. PCB stands for physicochemical and biopharmaceutical. It includes pKa, logP, logD, permeability through various tissues, solubility in various fluids, some pharmacokinetic parameters. There are transporter models. These are either substrate, non-substrate models or inhibition models. And there is a clearance mechanism model that predicts either metabolism, renal, or hepatic uptake. The metabolism module covers cytochrome P450, UGT, and as of version 9.5, aldehyde oxidase enzyme. We have substrate, non-substrate models for all of these. There are several isoforms of CYPs and UGTs. For the CYPs and for AOX, we have sites of metabolism models. So those predict where on a compound is metabolism likely to occur. For the 5 major CYPs, we have kinetic models. that predict Km, Vmax, and intrinsic clearance. For those same 5 major SIPs, we have inhibition models. And then finally, we have 2 models for total liver microsome clearance, a human model and a rat model. We have models for several toxicity endpoints. Cardiac toxicity includes the HERG. We have actually 2 HERG models. Liver, acute. Carcinogenicity includes the Ames models. And finally, sensitization and environmental toxicity. I covered HTPK just briefly in the last slide. One other prediction is called optimal dose, which is the dose required to achieve a target plasma concentration at steady state. Most of the ADMET predictor predictions are from QSAR models. So we begin with data that could be extracted from literature or acquired through a collaboration, etc. We characterize the compounds using our own proprietary molecular and atomic descriptors. And then we train a machine learning model using a method like artificial neural networks that I show here. One important point that I will come back to in a couple of slides is that we train not one model, but a collection of models. We call that an ensemble. And we find generally that ensemble models outperform individual models. This graph shows a performance chart for one of our models, the PKA model. The user manual has this kind of performance information for all of our models, as well as a description of the data that was used to build the model. Also, in the additional slide section and on our website, there is information about some independent third-party comparisons of different prediction software packages. And in particular, our PKA, logP, and solubility models have been shown to be best in class in those comparison studies. Now, the HTPK predictions are different in that they do not use QSAR models. They are the result of a mechanistic simulation of drug disposition. The model it's comprised of 2 components. There's the ACAT component that models the transit of a drug through the GI tract and the absorption into the enterocyte cells. And that is coupled with a 1-compartment PK simulation to model the clearance from the central compartment. So running this simulation involves integrating a system of time-dependent differential equations from which you can calculate parameters like fraction absorbed, fraction bioavailable, etc. Now this overall model is equivalent to a compartmental simulation in our widely used GastroPlus software. And it will give the same results, provided that you use the same input parameters. Those input parameters are things like compound permeability, solubility, PKA, etc. Those can be experimental values if you have them, or they can be predictions. The default is to use ADMET Predictor predictions for those inputs. If you happen to have your own experimental data and can build models on that data, then you can use predictions from those models as the inputs as well. And then this shows the predictive performance of the PKA, I'm sorry, of the HTPK module. We used all predicted inputs from ADMET Predictor and then estimated either fraction absorbed or fraction bioavailable for compounds from 2 well-known publications. Now, this is a much more difficult task, obviously, than predicting a property like logP or solubility. So we judge performance by the percentage of the compounds that are within either twofold or 1.5-fold of the experimental value. And these predictions are actually quite good, given the complexity of what we're trying to model. And you can often do better by using any experimental data that you might have. Okay, so that was a very, very brief introduction to ADMET Predictor. Now I will list some of the new features that we've added in this most recent version. The first is called structure sensitivity analysis. This is an interactive display that shows how the individual atoms of a chemical structure contribute to a predicted property. Another way of saying that is it allows you to see which regions of a compound are predicted to have the most influence on that property. And this is a screenshot of that window showing a single compound. I'm displaying the S plus log P model. The convention is for a green color to label atoms that are associated with a higher value of the property, and red color to associate with a low value of the property. So the hydrophobic atoms are colored green, the polar atoms colored red. I'll show a couple more interesting examples of this in the demonstration. So this feature was intended to complement an existing feature called descriptor sensitivity analysis. So that one gives information about how a change to a descriptor would influence a particular property. That information can sometimes be difficult to interpret because many of the descriptors used in ADMET Predictor are quite complicated and not easily mapped back to chemical structure. So this new feature was, at least in part, intended to address that limitation by being more chemically intuitive. The feature relies on a separate set of models that were built on a large reference data set using partial least squares regression with extended connectivity fingerprint keys, ECFP keys. So a new compound displayed in the window can be colored according to which ECFP keys it has and the PLS model coefficients of those keys. We have, I think, 21 total models that we distribute. They cover the PCB, metabolism, and toxicity modules, and users can also build their own models with their own data. And I will give a demonstration of that shortly. Another new feature we call regression uncertainty analysis. So regression model predictions, both our models and user models, now have accompanying uncertainty estimates. This complements an existing feature, which is the classification confidence estimates. The classification models are the yes-no type models or substrate, non-substrate. So those have had confidence estimates going back, I think, to ADMET Predictor version 7. So with this version, the regression model predictions will also have similar values associated with the predictions. The uncertainty estimates are computed from the standard deviation of predictions from individual models that make up the ensemble. I referenced the ensemble a couple slides back. ADMET Modeler by default includes 33 models in an ensemble model. Higher standard deviations of those predictions lead to higher uncertainty estimates and vice versa. The uncertainties themselves are estimates of the standard deviation of prediction errors. So they're telling you something about the errors. And hopefully the next slide will clarify that a bit. This shows the results of one of the validation studies that we conducted. We took a large solubility data set and divided the compounds into a training set and a test set. We built a neural network ensemble model using the training set and then used the model to predict the solubilities of the test compounds. We also generated uncertainty estimates for those test compounds. Now the graph on the left shows predicted versus observed solubility for the 200 test set compounds with the smallest predicted uncertainties. On the right are the 200 test set compounds with the largest predicted uncertainties. And what you see is that the compounds with the lower uncertainties actually have lower errors. The compounds are closer to the diagonal line. Whereas on the right, the compounds with the larger estimated errors, the larger estimated uncertainties also have larger errors. The points are more scattered. We have done a number of validation studies like this, and we consider it, to be a good validation of the overall approach. And lastly, I'll just say that the user manual will have a lot more mathematical detail behind the method, and it also places it in a more formal statistics framework. We've added some new models to version 9.5. There are 3 new transporter models. We have a BCRP substrate non-substrate model, OCT2 inhibition, and BSEP inhibition. We have a new Ames mutagenicity model that we built using data that we received from the Japanese National Institute of Health Sciences. And we have 2 models related to aldehyde oxidase, a substrate non-substrate model and a sites of metabolism model. We've also improved some of the existing models by adding new data. The blood-brain barrier penetration model, that's the classification model, human blood to plasma ratio, the rat plasma protein binding, and the rat liver microsome intrinsic clearance. We've expanded the metabolite prediction capabilities by now handling aldehyde oxidase, UGT, and esterases. These incorporate the corresponding substrate and site models when they are available. So for example, AOX, as I just mentioned, has both a substrate model and a site model. UGT, we have the existing 9 isoform specific substrate models. These also incorporate some custom rules derived from literature. And I will spend a little more time talking about this during the demonstration. We've made a lot of changes to MedChem Designer, which is the companion drawing or sketch tool that comes with ADMET Predictor. We've put a lot of the ADMET prediction functionality from Predictor into Designer. And that makes it very useful for experimenting with structure changes and then quickly predicting what effects those structure changes might have in terms of either ADMET or PK. So we've added the pKa microstates display, the atomic properties display. That's used for looking at atomic descriptors and some atom-specific metabolism properties. The logD and solubility versus pH curves. All the metabolite prediction features are in Designer, and a lot of the HTPK features as well. So FA, FB, some of the graphical features like CP time curves. And I'll show a lot of this in the demonstration. And then we have made improvements to the Pipeline Pilot and KNIME workflows. That was done through collaboration. For this release, we have focused on modernizing and simplifying the workflows, and that puts us in a good position to down the road add a lot more functionality to those workflows as well. There are many other enhancements that we've made, and hopefully I'll have a chance to show at least some of those during the demonstration portion. But for now, I will thank some of the individuals at Simulations Plus who were most directly involved with ADMET Predictor 9.5 development, and then just bring some attention to the 2 companies we worked with. One was Workflow Informatics, that was Chris Loden. He helped us with improvements to the Pipeline Pilot workflow. and then Nova Data Solutions. A guy named Mike Mazanets helped us a lot with the KNIME workflow. All right, so before I start the demonstration portion, I'm going to hand the controls back to Eric, who will conduct a short poll.
Yes, thank you, David, for this very nice overview. Indeed, I'm now going to run the first poll, and you will see it pop up on your screen, asking if you currently using ADMET Predictor. And I'm going to leave the poll up for about 30 seconds, after which I will hand the controls back to David. Well, thank you very much. I will now close the poll. Thank you for your feedback. And I will now hand all the controls over to David for some live examples with the software.
Okay, thank you, Eric. Okay, so for this part of the demonstration, I hope someone will chime in and tell me if my screen is not showing. Hearing nothing, I will continue. Okay, so for this part, I'm actually going to use the software and show some examples, see what we can fit in about 30 minutes. So first, I'd like to talk about the structure sensitivity analysis feature. I just have a few more examples to show. So I opened up a small SD file, and actually, rather than show this in ADMET Predictor, I'm going to send these compounds over to MedChem Designer, which I mentioned is the companion sketch tool. The structures are bigger here, they're easier to see, and this will actually allow me to talk a little more about some of the other functionality that we've added to Designer. So more and more Designer is incorporating the predictive capabilities of ADMET Predictor, which makes it very convenient to make structure modifications and then quickly predict what the effect of those changes might be. So for example, with these structures, you can just click the Add Me button and then quickly get all the same property predictions that you would see in the main ADMET Predictor window. As far as the structure sensitivity feature, that's available here in Designer as well as in Predictor. And I'll just show a couple of examples here. So this compound is called 3-nitrobenzanthrone. I read earlier that it has the highest ever observed positive score in the Ames mutagenicity assay. This is a compound found apparently in diesel exhaust. So I can send this compound to the SSA window. I showed the screenshot of this earlier. You first select a model. So we have about 21 models here. One of them is the new mute-NIHS model. So that's the Ames model built with the Japanese NIHS data. So if I show the compound, I first neglected to mention, we do predict that it's positive in all of our Ames models. And you see that the aromatic nitro group and the carbonyl group in particular are colored. Now, this is a classification model as opposed to a regression model. So there's a legend here indicating that green color is associated with a positive prediction. So in this case, Ames positive. There's a slider bar where you can sort of adjust the color intensity and see the relative importance of the various groups. And so the aromatic nitro stands out here, not surprisingly. You can also show the individual atomic scores themselves. That was a pretty easy example. This is an example, terfenadine, of a compound that it was a drug that was withdrawn due to cardiac toxicity. It's a known hERG blocker. our hERG models predict it to be a hERG blocker. If I show that in the window, I can select one of our hERG models. This is the classification model. And you can see it highlights the tertiary amine and some of the surrounding atoms, including the 2 atoms from the benzene rings here. The SAR around hERG blocking is a little bit complicated, but it certainly makes lots of reference to basic amines and to bulky nearby groups. So this seems to be a sensible prediction as well. I don't have time to show a whole lot of examples here. I wanted to show to highlight one other use of the structure sensitivity analysis feature, and that is that you can visualize arbitrary structure alerts. All right, so using structure alerts is fairly common. So this compound, trovafloxacin, is another compound that was withdrawn, this one because of liver toxicity. If I show that in the window, instead of selecting one of the existing models, I can also open up a file containing basically arbitrary structure alerts. Now we've put together one such file for this new release, so this will be distributed. And we created it by going through the literature and collecting alerts related to reactive metabolites. So I think there's something like 50 structural alerts contained in this file. It's just a flat file, and you can define your own or modify ours. And if I open it, any alerts from the file that hit the structure will be shown, kind of overlaid simultaneously, and then you can navigate over them. So this one says cyclopropylamine and Michael acceptor. Now, just to briefly mention, the same kind of file, like this file, can be used in ADMET Predictor as well. There you can append scores, like structure alert scores, to 1 million structures in the spreadsheet. But this window is a more graphical way of looking at the actual hits on the chemical structure. So that's one other use of the window. And then finally, I mentioned in the slides that you can build your own model to display in that window. So I'd like to just briefly show that process. For that, you need to open up a data set with structures and some property that you want to model. So I have a data set that I downloaded from Kemble. The data has PIC50 values against this receptor. IGF-1R is the insulin-like growth factor type 1 receptor. So if I open that file in Predictor, it has about 2,500 structures, all with PIC50 values. I can just briefly show the histogram of the activity values. So it's quite a range, up to about 10. To build this SSA model, there's a menu item for that called Create New SSA Model. You select a property to model. This could be activity, it could be an ADMET property, it could be anything. I'll keep the default option to build the PLS regression model, as I described in the slides. So this behind the scenes then is going to be generating the extended connectivity fingerprint keys and then building the partial least squares regression model. And then it saves it in a place where the window can find it. So I'll display just one compound in that window, and I'll sort and just choose the most active compound in the data set, which is this one. So now if I launch that window, this time from Predictor, the list of models will have the new model that you just built. And if I display it, this is what it looks like for this compound. There's a lot of green here, and this was the most active compound in the data set. You can kind of see that this portion of the compound is flagged as being most correlated with the high activity of the compound. Now, Since we have all of the data and the structures in the data inside Predictor, I can take a minute to just show some of the cheminformatics functionality of ADMET Predictor. And I think that will help kind of rationalize the color pattern that you see here on this compound. We can actually look for SAR by using the cheminformatics functionality in ADMET Predictor. So I think that'll be instructive to do just in a couple of minutes. One of the things that is pretty common to do is to organize a set of compounds into chemical families or classes. So that's one of the features of ADMET Predictor. There's a few methods to do that. I'll just keep the default option, a method called frameworks. Basically, it's ring systems. Technically, it's the Merco assemblies method from that Merco and Bemis publication from maybe 10 years ago now. Once you've done that, it's a fast operation. You can activate the classes tab, where now each row of the spreadsheet represents a whole group of compounds, a class of compounds, rather than just one. If I magnify this first row, you can see the blue highlighting indicates the scaffold for the class. So all the compounds in the class share that scaffold. The compound here is just the lowest molecular weight compound in the class. This column shows there are 534 compounds in the class. This is the molecular weight distribution. I can change it to the activity distribution. So it's a high activity class. You can also highlight the class and then show the relative activity. So the red indicates the activity of the compounds in the class. The blue are the compounds not in the class. So it has relatively high activity. And then just from the scaffold itself, the pyrazine ring and the pyrimidine ring, you see that the most active compound has that scaffold. So the most active compound is actually in this class as well. Another useful feature for looking at SAR is to build an R table. So that's a simple operation in ADMET Predictor. So you see there are 5 points of substitution around that scaffold. That's a little bit complicated, but there's a feature where you can search for subclasses, large subclasses within the class that simplify this R table a bit. It's kind of a powerful feature that would take me a little while to fully explain, but the default operation is just to search for a large subclass that simplifies this R table by removing some of these R groups. And it turns out there is such a subclass within this Chembl dataset. So just by clicking OK, it discovers a subclass of this class, a large one. It has 489 compounds. And here, basically, we fixed this position to be methyl and these two positions to be hydrogen. So most of the compounds have methyl here and hydrogen here. Our most active compound has that exact same substitution pattern. So this is a much simpler R table. We're only varying the groups out here and out here at positions 3 and 4, or 4 and 5, which are now 1 and 2. With just 2 R groups, it's pretty easy to do some further analysis. It's pretty common to display all of the R1 substituents against all the R2 as a matrix. And there's a feature to do that called the R group analyzer, where you can display the R1 by R2 matrix. So here's the scaffold with the R1 and R2. These are all the observed R1 substituents as the rows, and all the observed R2s as the columns. The points in the graph or in the matrix indicate compounds we actually have. They're colored by the PIC50 against the enzyme, and green indicates most active. You can sort that to put the actives at the upper left and the inactives down here. And you can see right away that there are 2 R groups that kind of stand out, these rows here, or this row and this column. If I look at that R1, it has that benzene pyrazine group, which is exactly what we see in this most active compound. And then at the R2 position, something a little different. It's the same aminopyrazole, but here with the ethyl substituent. And here we actually have the same aminopyrazole, but and actually the same ethyl, but here with the extra piperazine ring, which is actually colored a little bit less green. Now, I've spent a little more time with this, but I don't think there's time to show much more. I just wanted to give a little demonstration of some of the cheminformatics capabilities of ADMET Predictor, and hopefully that kind of explains what's going on under the hood in terms of producing this particular color pattern that you see. And hopefully now you have some appreciation of some of the capabilities of Predictor other than just property generation. Okay, I'll stop there with SSA, and I'm going to go back to Designer. I'd like to just spend a minute now talking about some of the HTPK features that we've added to Designer. So I'm going to open up a different data set. Actually, it's just one compound. So this is a compound that I took from a 2010 MedChem paper on the discovery of JAK kinase inhibitors. The structure was one of the initial leads that had a high potency against the JAK enzyme, but that had a very short half-life. And the paper described an effort to find a compound that was both highly active, selective also, but that was metabolically stable. And the focus in the paper in particular was on varying this side chain here. So activity was determined to be kind of governed by this region, but this was the side chain that could be varied to improve the PK. And I'll just adapt this a little bit to talk about trying to improve bioavailability, okay, just so that I can talk about the HTPK feature. So I'm modifying this just a bit, but so I mentioned that Designer now has a lot of the HTPK capabilities from Predictor. So if I click this button here, you'd see the same interface that you would see in ADMET Predictor, where you choose a species, you can enter one or more doses. We're predicting fraction absorbed and fraction bioavailable. I kind of mentioned that there's a lot of input parameters, and you can leave the default values, which are just ADMET Predictor predictions, or if you have experimental data, you can enter those as well. I'll just run it with default settings. And you see that the compound has a high predicted fraction absorbed, but just a moderate fraction bioavailable. So we can imagine that we are interested in making changes to this side chain so as to increase the predicted bioavailability. So in Designer, you can just copy the structure and then use the sketch tools to explore different types of groups out here. Another feature that I'll just show briefly is using ADMET Predictor to actually create a virtual library for you. So you can just create an R group here and then copy this into the clipboard, bring it back over to Predictor. And there's a feature to, it's called R-list explosion, where you can paste a scaffold with at least one R group and then either draw or load in some previously defined sort of substituents. So I'll load some that I created previously. There's 300 of them. I can show the first 100 in Designer. Designer has kind of a limited capacity, but I can show the first 100. They're all small substituents, and these were created using either the WDI or an FDA drug list. Like, I forget exactly how I created it, but they're drug-like fragments. Predictor will then explode out that virtual library and put the compounds in a new spreadsheet. There's 300 of them. You can now, for example, calculate the ADMET properties of these compounds. So this, again, I mentioned this, the PhysChem, the metabolism, toxicity properties, as well as the modeling descriptors. They all become new columns in the spreadsheet. You can run the same HTPK feature in ADMET Predictor. So we just saw this interface in Designer. I'll use the same parameters and just run that. So this is performing that, mechanistic simulation that I described for all 300 compounds. And then it places those predictions in the spreadsheet, the FA, FB, and some PK parameters. I'll just show the distribution of predicted bioavailabilities in the histogram. There's a large range from about 25 to 96%. And I'll just sort to find the highest predicted bioavailability and just copy it back over to Designer so we can see them side by side. So you see now the cyano group out here in the side chain. So I can rerun the simulation just to get the numbers side by side. So you see the predicted bioavailability of this compound is much higher. We predict 96%. To kind of get some type of explanation for why the predicted bioavailability is higher, there's another new feature in Designer that I'll show. That's the atomic properties window. So this was an existing predictor feature that we've just put into Designer. There's a lot of things you can display here. One of them is the sum of CYP clearance values. So this is for the 5 major CYPs. So you can see that there are 2 predicted sites of metabolism out on that side chain, and then 3 additional sites in the scaffold. Now, if I show the second compound with the cyano group, you've lost the sites on that group. And actually, the predicted metabolism rate is less for this compound, which could be explained by the lower logP of this compound. And that was one of the things that was mentioned in the original journal article. It was about trying to reduce the logP of the initial lead structures. It turns out this is the compound that was decided upon to move forward in that journal article. This is a known or a marketed drug now. It's called tofacitinib or Xeljanz. Now I searched for the reported bioavailability of this compound, and it's more like 75%, whereas we're predicting 96%. So our prediction is almost certainly high, although not bad, by the standards that we were discussing in the slides, all predicted inputs here. It's actually pretty good. If we were interested in, fully analyzing this, we would be thinking about things like, UGT metabolism, which we don't currently handle in the HTPK, metabolism that is not taking place in the liver, gut metabolism, for example. We do not handle that in the HTPK either currently. But you can hopefully see that having this kind of, quick ballpark estimate of these PK properties is useful, particularly in software like Designer, where you can so easily experiment with structural changes. Okay, so that I think is all I wanted to say about the PK. So the last thing I wanted to show was metabolite prediction. So I have just a few examples to show here. I mentioned that predicting metabolites is a feature both in ADMET Predictor and in Designer. In Predictor, you would typically run this on a large number of compounds, and the metabolites are displayed in the spreadsheet. Here in Designer, you select one compound at a time, but the predicted metabolites are displayed in a nicer, more graphical tree-like structure. So there's kind of 2 different workflows. Now I'd like to just spend a minute kind of reviewing the process of metabolite prediction. I think it will help. I'll be thinking about this compound, aripiprazole, and I'm going to focus on cytochrome P450 metabolite prediction first before I talk about the others. So when you predict metabolites, the first step is to send this compound essentially to the models for the 9 different CYP isoforms. So we have 9 substrate, non-substrate models for 9 different CYP isoforms. For each isoform that's predicted to metabolize this compound, we predict which sites on the compound are likely to be the site of attack. Each of those sites could yield a unique metabolite, which initially is going to be just the hydroxylated structure. But then that hydroxylated structure is processed by a set of rules that may lead to a spontaneous transformation to a more stable observed metabolite. So it's kind of a three-step process. And I'll just illustrate it graphically here for the case of SIP. So again, this is aripiprazole. This is the result. Here's the original compound. There are arrows indicating the metabolites. They're labeled by the responsible CYPs, of course, the predicted CYPs. You can see that the rules, the expert rules, are somewhat complicated. There are some cases where it's just hydroxylation. We have hydroxylation followed by a spontaneous transformation to a more stable tautomeric form. There's a case of N-dealkylation that here is producing two distinct fragments. So that is all contained in a customizable set of rules. You also see that at least for the 5 major CYPs, we have kinetic models. So we're able to provide estimates of the clearance rate and the yield on the metabolites. And then lastly, I just wanted to remember to say that this is not just a picture. These are, live structures that you can edit, and you can also predict the properties of these metabolites. So these are just like any other structures in Designer. Okay, now I'm going to go back. Actually, I'll just close this. So for the new features, which is AOX, UGT, and esterase metabolism, I have a couple examples to show here. I think I still have a couple minutes left. I first want to point out that for AOX, for aldehyde oxidase, we've grouped it together with the 9 CYP isoforms to kind of form a phase 1 metabolite prediction feature. And then we have something similar for phase 2 with the 9 UGTs. And if you are interested in just seeing, a particular either isoform or AOX, you can just, deselect them, and then temporarily they'll be disabled in the interface. So that sometimes cleans things up a bit. So just briefly, I'll just show these features. This is a compound that entered phase 1 clinical trials and that failed in the trial due specifically to AOX metabolism. The metabolite led to, I believe it was kidney failure. So now if I expand this button, you see that the default option is actually now called CYP/AOX. So if I select that now, you'll see the predicted metabolite. This is actually the observed metabolite. You see that it does also include the same rules that describe the spontaneous transformation to the more stable pyridone structure here. And I'll point out that we don't at this time have an AOX kinetic model. So we're not able yet to predict clearance rate or yield. So this compound is the primary phase 1 metabolite of midazolam. It is called 1-hydroxymidazolam. It is glucuronidated, and I can just show that. Another option on this drop-down is for UGT. So these are the 2 observed metabolites of this structure. You see that we support glucuronidation of both oxygen atoms, the hydroxyl case, as well as nitrogens. Not every exposed hydroxyl is going to be glucuronidated. I mentioned that we have developed a set of rules that was based on a review of literature. So it's not just a simple a process of glucuronidating every single exposed hydroxyl group. And then like with the AOX, we don't yet have kinetic models, so we don't predict rates or yields at this time. And then finally, this is the prodrug of aripiprazole. So it undergoes ester hydrolysis. So you can run that feature to see the final metabolite. It is also based on a customizable set of rules. So post-hydrolysis, there's actually the removal of this hydroxymethyl group here, just to produce the final active drug. You can, of course, modify those rules. Okay, so let's see. I think we had agreed that at about 10 minutes to the hour, we would try to wrap up this demonstration. So I do have more things I could show. I didn't have a chance to show the new modeler, which now allows you to see error bars since we have the uncertainty estimates. There are a lot of other nice features that we've added, but so that we can save some time for questions, I think I am going to hand the controls back over to Eric at this time to kind of wrap things up. I think do a final poll and then take some questions.
Yeah. Yes, thank you. Thank you, David, for these very nice examples. So before we jump into the Q&A, I would like to run the second poll. And I will launch the poll now. So based on what you heard today, how likely are you to evaluate or seek more information about ADMET Predictor? And I will let the poll run for about 30 seconds. We still have more people voting, so we'll keep it up for a few more seconds. And okay, so thank you for your votes. I'm now going to close this poll. Thank you. So we're now going to open the floor for questions. If you would like to ask a question using your telephone, please use the hand raising icon on your control panel and be sure to enter your unique audio pin, or you can enter your question in the questions pane of your control panel. As a matter of fact, I see that we already have one question which has come up. Is the structure sensitivity analysis enhancement in the base version of the software, or is it an extra module? We can answer that.
I'll answer that.
Yeah, David.
Yeah, so it is a base feature in the sense that, let's see, I think that the one model that's included that doesn't require any license is the S plus logP model. And of course, the thing I showed about the structure alerts and building your own models to display in that window, that doesn't require any license at all. The individual models that I showed, like solubility, permeability, the hERG binding, those models are tied to the corresponding license that you're used to from ADMET Predictor. So things like the Ames model, for instance, would require a toxicity module license, and the solubility, permeability models would require a PCB license. But the S plus logP comes for free, and the ability to show either structure alerts or build your own models, that doesn't require any new license.
Okay, thank you. We also have another question for MedChem designer. Can we run a chemical through multiple enzyme models at the same time? So CYP, esterases and UGT, for examples, and predict which would be the predominant pathway?
That is a good question. We don't now have the feature to run them in sequence. So The workflow now would usually be to first, well, I guess, do the esterases first if that's relevant, since that's kind of a pre-phase one. Then run the CYP AOX, and then on selected phase one metabolites, run the UGT feature. The part about the dominant pathway, is a little challenging at this point since we don't actually have quantitative models for the for either AOX or UGT. We do have it for the 5 major CYPs. So that's something we're going to continue to work on. But for now, it's kind of a one-by-one manual operation as opposed to having it all put together in sequence automatically.
Okay. Great. Thank you, David. Can you also tell us when we expect this new version to be released?
I hope I can. The plan is to have the official release be 2 weeks from this Friday, and that's, I believe, April 12th. We are certainly on track for that. We are writing the documentation as we speak.
Okay, excellent. Well, thank you. So I think we're just a few minutes from the end of the scheduled time. This concludes today's webinar. If you missed any part of today's presentation, the playback will be available at our website, www.simulations-plus.com. Also, if you want to meet with us, we are going to be at the ACS next week in Orlando, and also at the CHI Drug Discovery Chemistry in San Diego the week after. And I think you can see on that slide our presence at both of these conferences. We thank you for joining us today, and we wish you a wonderful rest of the day.